Assalamu alaikum. This is our last lecture. We're done. Well, almost done. So previously we talked about protein purification, and let's say that now we have pure protein. How can we analyze this protein in terms of molecular weight, characteristics, isoelectric point, etc. So there are a number of techniques that we'll cover in this lecture, inshallah. Okay, so the first one is known as gel electrophoresis. We talked about gel electrophoresis before in the molecular biology part of the cell biology and molecular biology course. And it's basically a method by which we can separate molecules based on size through a gel, right? If you remember that. So basically phoresis is like movement, electrical field. So gel is like a jello, and it's basically we separate proteins as they move through a gel as a result of an electrical field. So that's electrophoresis. Okay, so you can go back or you can watch this YouTube video to learn more about it, to see how it really works. So the idea here is that we make a gel, and we make a gel from two molecules, well, number of molecules, but basically the basic molecules are SDS, and the other one is polyacrylamide gel electrophoresis. So SDS is sodium dodecyl sulfate. It is a detergent, and I'll talk about it now in a second. And the gel is basically made of a polyacrylamide. So it is a chemical that forms sort of like a network as shown in here. Okay, so it's called SDS-PAGE as well. Now, so what we have in here is like two plates, and in the middle there is space, really thin space, and the gel is poured inside this space. So we have something vertical. Now we place samples here, right here, in what we call a well, right? So we have multiple wells, so we can put sample 1 here, sample 2 here, sample 3, and so forth. So we place these samples inside the gels, and we allow protein molecules in a sample to move through the gel. Now notice that movement occurs from the cathode to the anode, okay, because we make proteins all negatively charged, as I'll show you in a second. So proteins move through this network, this mesh right here, okay? Now logically, as you would probably know, that smaller proteins would move faster than larger proteins, okay, because now the spaces, or let's say that, well, the spaces allow for smaller molecules, smaller proteins to move through them, versus larger molecules that would have a hard time moving. Now notice that inside a well right here we have a protein sample, and this protein sample, let's say that it contains 100,000 different protein types, and we have tens to thousands of molecules of each type. So for example, in yellow here there is one type, okay, and there are multiple molecules of this type. Same thing with the green balls and the purple ones. Okay, so all of these proteins now, protein molecules, they move at the same speed together, right? So, and they form what is known as a band. Okay, they move at the same speed because they have the same molecular weight, right? Okay, so then we take this gel and we stain the proteins. We color the proteins with a certain color, certain chemical, and each protein would look like a band, as you can see in here. So in this sample right here, so sample 1, there is nothing, no proteins whatsoever. So you look at sample 2, there are multiple bands. Now remember that each band may represent a type of protein molecule. Of course, each band contains many molecules of the same same molecule, same protein. Okay, so each band may represent one protein type, or maybe multiple protein types, because they have the same molecular weight, so they travel at the same speed. So each band would contain like a mix of different protein molecules all having the same molecular weight. Okay, so you can look at these bands and say, aha, like this is more intense than this one right here. So you said it means that it seems that there are more of this protein inside my sample versus this protein right here. Okay, now movement occurs downward according to molecular weight, according to size. Notice that now these are the large molecular weight proteins and these are the small molecular weight proteins. Now, notice that sample 2 is very similar to sample 6. If you look at the pattern of protein banding, they almost look identical. Now, notice these two samples right here, okay, number 6 and 7. You can see two bands. They are running very close to each other, but this one right here is a bit larger than that one here. You see? Okay, so that's the whole idea of SDS-PAGE. So what is the gel made of? It's made of a molecule, a chemical known as acrylamide. And this acrylamide is a small molecule, but it has the ability to form polymers. Okay, so a reaction is initiated by which this acrylamide molecule right here would form polymers. Now, these different polymers are then connected to each other by another molecule known as bisacrylamide or methylene bisacrylamide. We call it in the lab bisacrylamide. Okay, so these different polymers are connected to each other via this bisacrylamide, forming a network of fibers or polymers that would allow proteins to be separated from each other according to size again. Now, so why do we add SDS? So SDS is an anionic molecule, right? It's a detergent. It contains both a hydrocarbon chain, like a fatty acid, okay, and it contains a charged group at the end. Now this group is negatively charged. Now what happens is that when we add SDS to a protein, and this protein molecule, it has a certain structure, right? And this structure is made of non-covalent interactions. Now, when we add SDS, what SDS does is that it inserts itself inside the protein. So it's sort of like it coats it and it denatures it. So it breaks the non-covalent interactions. Now, so the protein loses its structure. It gets denatured. And SDS coats the protein, so the protein molecule now is totally negatively charged. Now, all protein molecules then would have this. They would all be denatured, and they would all have negatively charged, or they would all have negative charges. Okay, so they are uniform in that sense. And this is why proteins move from the cathode to the anode, okay, from the negative side to the positive side because of that. So SDS has two purposes. One, it denatures proteins. Two, it unifies the charge on all proteins, making them all negatively charged. Now, except that proteins, not only that they have these non-covalent interactions, okay, especially for single subunit proteins, monomeric proteins, they may also have disulfide bonds. Now SDS cannot break these disulfide bonds. Okay, so we need something to break them up, and what we need is a reducing agent. So a reducing agent like beta-mercaptoethanol or dithiothiotol, okay, these can be added to proteins breaking these disulfide bonds. Okay. Now, if we add SDS, of course, all proteins would then be denatured and separated from each other as well. Now, proteins are added inside these wells. We apply electricity. Here we have the cathode, the anode, and proteins then again they move through the gel and they get separated according to size. Okay, and then we stain them afterwards, and you can see each band right here again representing a group of proteins all having the same size. They can be of the same protein type or different protein types. So here showing SDS-PAGE of different samples. In the homogeneous right here, the homogeneous means that it's basically all proteins in a sample. So let's say that we have a group of cells, whatever, epithelial cells, intestinal cells, brain cells, whatever. We break them up and we release all proteins. So we homogenize these cells or tissue or whatever, sample. And then we separate all proteins in a sample. So we have many different types of proteins. We have large proteins and small proteins. We have nuclear proteins. We have cytoplasmic proteins. We have positively charged proteins. We have negatively charged proteins. Okay, but they're all when we do SDS-PAGE, they are all negatively charged, right? But here, that's in case we do salt fractionation. You know, salting in, salting out. That idea, we can get some protein purification, but it's really not that clean. It's quite dirty. That's in case we do ion exchange chromatography. Okay, again, it's it's fine, but it's not the best. That's if we do the molecular exclusion chromatography. Notice that here in a certain fraction we collect proteins of similar sizes. So here we eliminate the small molecular weight proteins and the large molecular weight proteins as well. Affinity chromatography is the best, right? Because using affinity chromatography, we can only isolate a single protein because binding is very specific. So that's the idea of SDS-PAGE. Okay, so we analyze our samples. Now, let's do these problems together. So, describe the protein's structure based on the following results of SDS-PAGE. Okay, so the idea here is that we have a sample that contains a single protein molecule. Okay, this protein molecule can be monomeric or polymeric. It can be small or large, whatever. It can have disulfide bonds or it may not have disulfide bonds. So what we want to do is describe the structure of these different proteins after we analyze them by SDS-PAGE and we see the banding pattern. Okay, so let's go into the first one right here. All right, so the problem says under non-reducing conditions. What do we mean by non-reducing? It means that we have not added the reducing agent, whether it's beta-mercaptoethanol or dithiothiotol. These are the reagents or chemicals that break up disulfide bonds. They reduce disulfide bonds. So whether these disulfide bonds occur within a monomeric protein or a protein with a quaternary structure having multiple polypeptides. What we do here is that the disulfide bonds are maintained. They're not broken up yet. Under non-reducing, denaturing. What do we mean by denaturing? It means that SDS is added. So all proteins are uniformly negatively charged. They all have lost their structure, tertiary structure. So migration of the proteins depends on their size, not necessarily anything else like shape. Okay. So a protein exists as as a 40 kilodalton band. So right here we have only one band under non-reducing conditions. Okay. Now, however, under reducing conditions, the protein exists as a single band with a size of 20 kilodalton as well. Okay, so what happens here? So the protein, we know that since there is a change in size, we know that there is a disulfide bond. Okay, now the size has been reduced from 40 kilodalton to 20 kilodalton. What does that mean? It means, it seems that the protein is composed of two polypeptide chains. Each one is composed of 20 kilodalton. So when they are connected to each other via disulfide bonds. So when these two polypeptide chains are connected to each other under non-reducing conditions, they travel as one single unit with a size of 40 kilodalton. However, when we break up these disulfide bonds, each polypeptide chain moves separately, but since both of them have the same size, they travel together as 20 kilodalton bands. Okay, remember that each band contains many protein molecules of the same size. Okay, so we're not talking about a single polypeptide chain. Okay, so how can we describe this protein? I would say this is a protein that is made of two exactly the same polypeptide chain. It's just an assumption. So I say that the protein is a homodimer, right? We say that this protein is a homodimer. Great. So I hope you got the idea. So let's get into the second problem. So under non-reducing, again, without any reducing agent, denaturing conditions, a protein exists as two bands, right here. Sizes 30 kilodalton and 20 kilodalton. Now, on the other hand, under reducing conditions, the protein exists as two bands as well, except that the sizes are different, 15 and 10. Okay, so how can you describe this protein? Well, note the change in size, right? So it was 30 and 20, now it's 15 and 10. So you know that there's at least one disulfide bond, okay? So it seems that this 30 is composed of two polypeptide chains. Each one has a size of 15 kilodalton. So 15 and 15, that's 30. So, and these two are connected to each other via disulfide bonds. And it seems that this 20 kilodalton band is basically two polypeptide chains of size of 10 kilodalton. So these two are connected to each other via disulfide bonds, that's 20. Great. And it seems that these two are connected to these two using or via non-covalent interactions. So it seems that this is a heterotetramer, just like hemoglobin, okay? A heterotetramer, right? Two polypeptides seem to be the same, 15 and 15, and these two seem also to be the same, 10 and 10. Just like hemoglobin, two alphas, two betas, and the two alphas are identical and the two betas are identical as well. Okay. Here's the third one. Now, under non-reducing conditions, a protein exists as two bands, 40 and 20. Great. Under reducing conditions now, there's only one band of a size of 20 kilodalton. Again, there is a change in size, so there is a disulfide bond, at least one. So, how can you describe this protein? So it seems that this 40 is actually two 20s, right? So 20 and 20, and you have a third 20 right here. So I would say that this is a heterotrimer composed of 3 polypeptide chains, and two of them are connected to each other via disulfide bonds, and these 2 dimers are connected to this polypeptide chain via non-covalent interactions. Okay, and I would assume that it is a heterotrimer, that these 3 polypeptide chains are different from each other. Okay, it's just an assumption. Of course, more work is needed. So I hope you get the idea. Now, there is another technique known as isoelectric focusing. And the idea here is that proteins are separated not based on size, but rather based on their isoelectric point. So, what is isoelectric point? It is the pH when a protein is charged, but the total net charge is equal to 0. So it's neither negatively charged nor positively charged. So what happens here is that a gel is created, okay? But this gel actually has a pH gradient, okay? Meaning that right here we have low pH, let's say starting at pH 3. So we have here 3.1, 3.2, 3.3, 3.4, 3.5, etc. Here we have pH 7, 7.1, 7.2, 7.3, etc. Here we have pH of 9, 9.1, 9.2, 9.3, etc. all the way to 10. So we have a range, a gradient of pH from pH 3 all the way to pH 9, and these are fixed. Okay. Now we add proteins to the gel, and these proteins, without any SDS, of course, so these proteins have their own isoelectric point or their own charge. Now, and we apply electricity. So proteins move to where the pH matches their isoelectric point. When a protein fits, or when a protein is located exactly at the pH that is equal to its isoelectric point, it's not charged. And as a result, it doesn't move to either side. That's it. It's neutral. It doesn't move here. It's not attracted to the cathode nor to the anode. Okay? So it sits at a certain position. That is at a pH that is equal to its isoelectric point. So proteins here are separated according to charge or according to their isoelectric point. So let's look at this animation right here for isoelectric focusing. So again, we have a strip, a gel strip, with pH gradient from pH 3 all the way to pH 10. We place proteins inside this gel, and we apply electricity, and proteins move, and they stop once they reach the pH that is equal to their isoelectric point. Okay, so here we have proteins separated based on charge or isoelectric point. This protein has low isoelectric point. This protein has intermediate isoelectric point, and this protein has high isoelectric protein, or in other words, it is a basic protein. So someone came up with an idea, and he said, well, why don't we combine both techniques, that is isoelectric focusing and SDS-PAGE? Why don't we first separate proteins based on charge, and then we separate them based on size? And this technique is now called two-dimensional gel electrophoresis. So the idea here is that we can separate proteins that have the same size but different isoelectric point, and we can also separate proteins that have the same isoelectric point but different sizes. Okay, so that gives us better resolution, okay, in terms of protein separation. This is a real two-dimensional gel electrophoresis where every spot may represent a single protein type. Of course, again, every spot represents thousands to millions of protein molecules. They can be of the same type, or they can be of different types, but they have same isoelectric point and size. So these two proteins have the same size, but they have different isoelectric points. Okay, and these two proteins right here, they have the same isoelectric point, but they have different sizes. This one here is smaller than that one. Okay, so let's look at the animation again, combining both. First, we have proteins separated based on their isoelectric point, and then they get separated based on size. Okay, so you can see again this protein is an acidic protein with sort of like a low molecular weight. This is a more of a basic protein, but having is a large molecular weight. Okay, and so on. Now, people have then developed something known as immunoblotting or Western blotting. Now, the idea was really more like a joke. So a scientist named Southern made Southern blotting for DNA, right? And then people in Chicago, they came up with a technique for RNA analysis, and they called it Northern blotting, since that's in the northern US. And then people from California came up with a similar technique, but for proteins, and they were located in California, and that's why they called it Western blotting. Okay, so the idea here with immunoblotting is that we want to detect specific proteins or a specific protein following SDS-PAGE. So first, proteins are separated in a gel electrophoresis. So right here you have three samples, three completely different samples having different protein profile, okay? Different composition of proteins. So proteins are separated. That's one, step number one. Step two, now the proteins are transferred from a gel to a special membrane. Remember that piece of paper that I told you about? So proteins are now located on the membrane at the exact same position as they were located on the gel, right? So you would have the large molecular weight proteins would be here, they would transfer right there, exactly at this, it's parallel to where they're localized in the gel. Small molecular weight proteins would also transfer to the membrane. So the membrane would look exactly like how proteins were organized or separated on a gel. Then what we do is that we take this membrane, and instead of adding a probe as in southern or northern blotting, we add an antibody, and this antibody is called a primary antibody. It targets a specific protein. Okay, so you have all of these proteins all around the membrane, but the antibody, since it's very specific, it binds to a single protein molecule. And then this primary antibody is targeted by a secondary antibody, and this secondary antibody would emit a signal. It would produce a signal. Okay, so this signal would appear on the membrane, right? Because that's enlarged. Okay, so it would look something like this. You see only a single band. Okay, and it's this one right here. So instead of seeing all of these different proteins, we only see a single one. Okay, so this is the protein of interest. So I know that these three samples have the same protein. They express that protein. Okay, so you can go to the YouTube and watch this video if you want. So that's immunoblotting. Well, we can use antibodies for another technique, and this technique is known as ELISA. Okay, it's not ELISA, it's ELISA. which is an abbreviation for enzyme-linked immunosorbent assay. And the idea here is that we want to detect a certain protein in a sample, okay, but without separating the proteins like we did with the SDS-PAGE. Okay, and this technique is very important, and it's used in clinical laboratories for disease diagnosis, detection of hormones, let's say, detection of certain proteins. It can be used to detect plasma proteins, viral proteins like the coronavirus, for example. So it can be used for different purposes. And the idea here is that we would have a solid support, okay? And we would add the antibody known as a capture antibody. It's like a primary antibody, okay? And it is attached to that solid support. Then we add our sample. Our sample would contain thousands of millions of different protein molecules. If the protein that is targeted, that can be recognized by the antibody, is there, then the antibody would capture it. It would bind to that protein. And then we can add another antibody that targets the same protein but at a different epitope. Remember what epitope is? But a different epitope, and this antibody emits a signal. Okay. Now remember, the solid support would have the same antibody, but there would be a lot of it on the solid support. So each one of them would capture the same protein. Okay, so you will have a strong signal if the protein exists at a large quantity. Now, the thing is, if the protein does not exist, it means that there's nothing that would bind to this antibody. There would be no binding of this other antibody to that protein. There's nothing to bind to, and as a result, there would be no signal. If there is a lot of this protein, then a lot of it would bind to the antibodies that are attached to the solid support, and the signal would be very strong. Now, if there's little antibody, if there's little antigen, little of this protein or molecule in a sample, then the signal would be weak. So the intensity of the signal tells me how much of this molecule exists in a sample. So here's the idea again. We have a solid support. We have this many of these antibodies on the solid support. Okay. We add our sample, virus, antigen, whatever, plasma, whatever sample, we add it. If the antigen exists, it would bind to the antibody. And then we target the same sample with another antibody that would bind to a different epitope, and a signal is emitted. Okay. Right here we have a negative reaction, meaning that the sample does not contain the antigen, meaning that if we add the antibody, if you add the sample, there's nothing to bind to this antibody. If we add the other antibody, again, there's nothing to bind to, and there would be no signal. Okay, and the intensity of the signal tells me how much of that antigen exists in my sample. So this is how it looks like. This is a plate. There are different wells. We can add the samples to these different wells. So we have sample 1, sample 2, sample 3, sample 4, etc. Okay. Now notice here, for example, we have very weak signal in here. Okay, meaning probably that there is nothing in there. There is no antigen. Here we have a strong signal. Okay, so compare this. So you can have gradient of colors depending on how much antigen or molecule there is in a sample. And the plate is like the size of your palm, by the way. So that's ELISA, very powerful technique. Okay, so let's get into something else. This is known as protein sequencing. The idea is the same as doing DNA sequencing. That is, we want to determine the order of nucleotides in DNA. So with protein sequencing, we want to determine the order of amino acids in a polypeptide or in a peptide. That is, in other words, I want to determine the primary structure of a peptide. So the basic technique for protein sequencing is known as Edman degradation. So scientists known as Edman came up with this technique, and the idea is that the amino, okay, and these amino acids, once they're removed, they are identified. And then the second amino acid would be removed and identified, a third amino acid would be removed, identified, and so on. So that's Edman degradation. And the technique is based on the use of a molecule known as phenyl isothiocyanate, PITC. What it does is that if we have a peptide in here and we add this PITC, it will bind to the first antibody at the N terminus, and it would remove it. It will take this and we can identify it using chromatography techniques. Okay, just like the ion exchange or size exclusion chromatography, whatever technique. So we identify this amino acid. Then we add PITC to the peptide again, and it removes the amino acid at the N terminus, which is now the second amino acid, which is now the first one that exists in that peptide, or that is present in this peptide, right? It gets removed and then identified by chromatography and so on. Okay, and this can be done 50 times, so we can determine the order of the first the first 50 amino acids in a peptide, which is good. So but what if the peptide is larger than that? Well, we need other techniques as well. So, or we can take a polypeptide and we can cleave it into smaller peptides, and we can identify the order of amino acids in these different peptides. So, there are other techniques by which we can determine the order of amino acids. And these techniques are based on cleavage methods, specific cleavage of peptides. And this cleavage can be done either chemically or enzymatically. So we can use endopeptidases or we can use exopeptidases. The difference is that exopeptidases cleave proteins at either end, at the N terminus or the C terminus. So we have two types of exopeptidases. We have amino exopeptidases, which cleave amino acids at the N terminus of peptides. And we have carboxy exopeptidases that cleave peptides at the carboxy terminus. And we have endopeptidases that cleave peptides within the peptide itself. Okay? Now, and this cleavage, by the way, is very specific at specific points. It doesn't occur randomly. So for chemical digestion, for example, we use a chemical known as cyanogen bromide. And cyanogen bromide cleaves peptides at the C end of methionine. So if we have a peptide and it contains within this peptide, it has methionine. If we add cyanogen bromide, this cyanogen bromide would cut right here at the C end, or at the C end of the methionine. Okay. So let's say that we have a peptide that has 10 methionine residues. This would yield 11 peptides, by the way. So, okay, mathematically, it means that 11 peptides would be generated. Now, let's say that I have a peptide. I know it contains 10. I know it contains methionines, but I don't know how many of them. And let's say that I have, when I add cyanogen bromide, I have 10 peptides generated plus a single amino acid. What does that mean? Well, it could mean that methionine is at the N terminus. Okay, so when it's cleaved, methionine is cleaved by itself, right? Or it could mean that this methionine could be the one before last. So the amino acid at the C terminus is cleaved by itself. Okay, think about it. Now, the thing with endopeptidases is that they cleave proteins at specific points. Okay, we talked about some of these endopeptidases before. So, trypsin, for example, it cleaves a polypeptide at the C-terminus of arginine and lysine residues. So if you have a peptide right here, and it contains a number of lysines and arginines. So here you have a lysine. Okay, so you have an arginine right here, you have a lysine right here, you have another lysine. Okay, let's go here. Here's another arginine, and so on. So if we add trypsin to this peptide, it would cleave right here, and right here, right here, and right there, generating 5 different polypeptides. Okay. These are the different endopeptidases and where they cleave. So trypsin cleaves peptide bonds at the C-terminus of arginine or lysine, but not if they are next to proline. Why? Because proline sort of like distorts the bond, the peptide bond, so it doesn't allow trypsin to cleave. Chymotrypsin, it cleaves at the C-terminus of phenylalanine, tyrosine, or tryptophan, but not if next to proline. Again, it's a troublemaker. Elastase, it cleaves at the C-terminus of alanine, glycine, serine, or valine. Again, not next to proline. Now, pepsin, the stomach peptidase, cleaves at the N-terminus of leucine, phenylalanine, tryptophan, or tyrosine, but not if next to proline. That is, if proline is involved in this peptide bond, that is, if it comes right before leucine, phenylalanine, tryptophan, or tyrosine, then pepsin cannot cleave this peptide bond. Okay? So you need to memorize this. Now exopeptidases, as I said, there are 2 types. We have amino exopeptidases or carboxy exopeptidases, depending on where they cleave. So let's look at this peptide right here. So we have this long peptide, and we cleave it by chymotrypsin or cyanogen bromide. Now I can ask you what different types of peptides generated. Okay, if chymotrypsin and cyanogen bromide are added separately, or if they are added together. So if they are added separately, chymotrypsin would cleave right after phenylalanine, tryptophan, or tyrosine, right? Now, cyanogen bromide is a chemical that cleaves at methionine residues. So it cleaves right here, it cleaves right here, and so on. Okay, so I can tell you that we have generated these different peptides. Can you put them together like a Lego? And I love that. So you can combine all of these different peptides and their sequences, and you can paste them into one large peptide. Okay. Now, here's a problem right here. So you have a sample of an unknown peptide, and it was divided into 2 aliquots. One aliquot was treated with trypsin, the other one with cyanogen bromide. Now, these are the peptides that are generated by trypsin and by cyanogen bromide. And now the question is, can you put them together into one large peptide? So again, you can see that you have asparagine, threonine, tryptophan, methionine, isoleucine, and lysine. Can you see these in here? Okay, So you can try to glue things together. So right here you have isoleucine and lysine. So you know that afterwards, after this, you have glycine, tyrosine, methionine. You have glycine, tyrosine, methionine, then glutamine, phenylalanine, and so on. So you put them together into one large peptide. Now, here's a sample, again cleaved with trypsin, and the same peptide was treated with chymotrypsin as well. And these different peptides, shorter peptides, were generated. Again, what is the original sequence of the peptide? Again, you put them together into one large peptide. Now, I can ask you questions like, you have a pentapeptide, that is a 5 amino acid peptide. And this, this peptide is cleaved by cyanogen bromide, generating, let's say, a dipeptide and a tripeptide. The dipeptide is negatively charged and the tripeptide is possibly charged. And if the same pentapeptide is cleaved by trypsin, then you get this type of peptide and that type of peptide, and so on. So basically, I can give you problems like that in the exam. You should be quick in solving the problem. I can ask you what the sequence is of this peptide. So what I'll do is give you sort of like a sheet that contains different types of problems and their solutions as well. So you can try to solve them yourself. So it's really important to determine protein structure. And the idea is that once we know the structure of a protein, we can understand how it functions, we can understand how it interacts with other proteins and other molecules, we can design drugs against this protein. Okay, so it's really important to understand protein structure and determine protein structure. And there are two ways. One way is known as crystallography, and the idea is that we change the protein we purify a protein, and then we crystallize it. We turn it into a crystal. So it looks something like this, and this can be done at really low temperature. Okay, and it takes time to crystallize a protein. And then we hit the protein, the crystal, we hit it with X-ray. Now the X-rays would get scattered as they hit the atoms. They get scattered, and they form something that looks like this in the background. And scientists, I don't know how they do this, but they translate this image into this beautiful structure right here. Okay, so this is really great. Now the problem with crystallography is that, one, you have to purify the protein. Okay, that's one. The other thing is that it's really hard to crystallize it. It's not really easy. So usually it gets frozen for a long time, few months maybe, in order for it to form a crystal. So if the electricity gets turned off, well, that's a problem really, because you have to start all over again. Now the other thing is that when you take a protein out of its natural environment, it will form a different structure, especially for membrane proteins. So these membrane proteins contain hydrophobic regions, right? Because they insert themselves into plasma membranes. So if you remove this protein out of its natural environment, out of the plasma membrane, now the structure that you will get is not really the natural normal structure of a protein. So as a result, scientists came up with another technique known as nuclear magnetic resonance spectroscopy. And the idea here is that this can be done, determination of protein structure can be done in solution rather than turning proteins into crystals. Okay, so you can really determine protein binding changes that happen as proteins bind to different molecules, like for example to oxygen. You can see this shift, the the movement of structure, the transition from the T state to the R state of hemoglobin. You can determine myoglobin structure, and you can do the same for the beta subunit of hemoglobin, and you can put them on top of each other to see differences in between beta globin protein and myoglobin, and so on. So the beauty of NMR is that, again, you can do it in solution. So really it can be done for sort of like the natural environment of proteins. And we're done.